Hello, General Biology, and welcome to the fourth in the screencast series of Chapter 9, Cellular Respiration. And we left off with the last one talking about glycolysis and the process of glycolysis. So just to give yourself a little bit of a refresher, because I don't know if you watched that last night and are now watching this another night, or if you're watching them back to back. So either way, this will be a little bit helpful to go over this. So remember, this is our glucose molecule, and that glucose molecule was made by the process of photosynthesis. And we're in glycolysis, we're in the cytoplasm of the cell, and we have to break down this glucose molecule into 2,3 carbon compounds in order for it to pass through the membrane of the mitochondria. And remember, glucose has carbon, hydrogen, and oxygen that are present in it. So we're going to be using some of the electrons and some of the hydrogen ions that are associated with the glucose molecule. So it takes 2 ATPs to start the chemical reaction. And 2 ATPs break down the glucose into 2 3-carbon compounds. What happens when ATP breaks down is we get 2 ADPs and we get independent phosphate groups, which is not pictured here, but you have to remember are actually there. So now we have the 2 3-carbon compounds. In this breakdown, we wound up getting a pair of electrons here and a pair of hydrogen ions here, a pair of electrons here, and a pair of hydrogen ions there. So we have an electron carrier, 2NAD+, which is a coenzyme. It's a helper enzyme that's going to help the reaction. It's going to pick up the electrons from here and the hydrogen ions from there and become NADH. It's going to pick up the electrons and hydrogen ions from here and become NADH. So we're going to take 2 NAD pluses to produce 2 NADHs, which will go off to the electron transport chain. When we pull those electrons out of the 3-carbon compounds, that's enough energy that is being released to take 4 ADP and join them with the independent phosphates to make 4 ATP. So when we look at our totals of ATP, we gained 4, but at the cost of 2. So when we take 4 minus 2, we actually gained 2 ATP from the process of glycolysis, as well as 2 pyruvic acids, because now these 2 3-carbon compounds that we get as a result are going to be pyruvic acids, and these pyruvic acids are going to be able to move through the membrane of the mitochondria, where we're going to go to next. Alright, so here we are with the Krebs cycle. And I just want you to take a deep breath. I know it looks really complicated, but it's not. You're going to hear me. I just want you to sit back and hear me talk about it. And if you have questions, highlight where you have questions, what you don't understand. And again, we're going to go over it in class. But this, I just want you to hear it, and I want you to hear me go through it. And I want you to watch carefully, so make sure that you're focused on it. So we are now in the mitochondria. The pyruvic acid just passed through the mitochondria. It passed through the outer membrane, and it passed through the membrane of the cristae, and it's now in the matrix, in the center part of the mitochondria. Now we're only looking at 1 pyruvic acid. We know that 2 are made from the process of glycolysis. So in essence, this cycle is going to happen twice for every 1 glucose molecule. But we're going to simplify it and just follow 1. And I'm going to try to write again, and you know that that's not a difficult thing for me to do with it, or not an easy thing for me to do with this. Okay, let's shift this. Can we shift this down a little bit? No, we can't. All right. So here we have our 3-carbon compound. And the minute it gets into the matrix, it's going to become unstable and it's going to break down. So it's going to break down between the last 2 carbons in the pyruvic acid. Now the carbon's not going to go by itself. It's going to take some oxygen. It's actually going to take 2 oxygen. So what happens to be released is a molecule of carbon dioxide, one of the carbons with 2 of the oxygens that were already present in the 3-carbon compound. So this is known as a byproduct, and I'm just going to write the letter B and P to represent byproduct. As we saw in glycolysis, whenever there's a breakdown, there's going to be release of hydrogen ions and electrons, which we're just going to represent by electrons. So when this when this carbon dioxide molecule breaks away, it's going to release 2, if this will let me write, it's going to release 2 electrons along with hydrogens, which we're not picturing there. And those electrons are going to be picked up by NAD+. Notice that there are negative charges, positive charges. They're going to be picked up by that. They'll become NADH, and this NADH is going to go off to the electron transport chain. So we'll see that NADH a little bit later. So now we've lost 1 carbon to carbon dioxide. We have a 2-carbon compound. Here's that 2-carbon compound. So it's just lost energy. Anytime it breaks off, it loses some energy. And it's going to need help in order to go through the next series of chemical reactions. So it's going to get help from an enzyme called coenzyme A. And I want you to remember back to the chapter where we talked about enzymes, and I pulled out my silly putty, and I said it just kind of molds around the substrate in order to help with the reaction. And that's exactly what we're going to see here. This coenzyme A is going to wrap itself around the 2-carbon compound that we see here and form a temporary compound known as acetyl coenzyme A. So that's this 2-carbon compound along with this helper enzyme coenzyme A to make acetyl coenzyme A. And now as acetyl coenzyme A, it's ready to enter a series of chemical reactions. So in the matrix is a 4-carbon compound. This 4-carbon compound is going to chemically join with acetyl coenzyme A to form a temporary 6-carbon compound known as citric acid. And one of the reasons why this is also referred to as the citric acid cycle. So citric acid in the matrix is unstable. One of these carbons is going to break away, and it's going to take with it oxygen. So we're going to get another carbon dioxide as a byproduct. And as you already know, anytime we get a breakup, we're going to get electrons that are going to be freed up. There's actually going to be 2 pairs of electrons along with their hydrogens that are going to be freed up. So those electrons, they're going to be picked up by NAD+, they're going to become NADH, and where do you think that's going to go? Same place this went to. This is going to go off to the electron transport chain. So off and away we go. All right, if we lose 1 carbon, we're going to have a 5-carbon compound left. So here is that 5-carbon compound. And guess what? it's going to continue to break down. And we're going to lose that carbon along with carbon dioxide, our byproduct, our oxygen, our byproduct, and that's going to be released from the cell. And again, we're going to get more electrons that are going to be released. Now we still have a pair of electrons from here that weren't picked up, and we're going to get 2 more pairs of electrons that are released from here. So we've got a couple of pairs of electrons that are available. They're going to be picked up, some of them are going to be picked up by NAD+ to form NADH, and where's that going to go? You guessed it, the electron transport chain. Now hopefully you're saying, Mrs. Saunders, every time we release electrons, isn't there energy being released? And yes, there is. That energy is going to be used to take ADP, join it with an independent phosphate group, and make ATP. So the Krebs cycle is giving us some ATP, which we need, as well as these electron carriers. All right, we lost a carbon here. We're down to 1, 2, 3, 4 carbon compound. Here it is. And we're going to have 2 pairs of electrons that I'm going to represent by single electrons here. 1 pair of electrons is going to be picked up by a new electron carrier called FAD. And it's the same as NADH. It's just a little bit different in its chemical makeup. It's still a coenzyme. It's going to form FADH2, and this FADH2 is going to go off to the electron transport chain. So that means we have 1 more pair of electrons, and they're going to be picked up by NAD+ to become NADH, and that's going to go off to the electron transport chain. So now this de-energized 4-carbon compound that remains will be ready for the next acetyl coenzyme A to enter the cycle, and that will happen all over again. Let's switch colors here for a minute. Let's do this bright green, and let's just see how many electron carriers we got from the series of energy extracting reactions. We got 1 NADH here. We've got 1 NADH here for 2. We've got 3, and we have 4. So we got 4 NADH from this reaction. Let's see how many carbon dioxides we got. It's still produced, but they're a byproduct. We've got 1, we've got 2, and we've got 3. They're going to be released from the cell, and that's the carbon dioxide that we exhale from our lungs. And then the last thing that we gained, is FADH2. Oh, no, that's not the last thing, is it? Oh, you're right. I heard you say it. But Mrs. Saunders, what about the ATP? Let's focus on the FADH2 first. Another energy carrier off to the electron transport chain. So we've got 1, 2, 3, 4 NADHs, 1, 2, 3 carbon dioxides, 1 FADH2, all from 1 pyruvic acid. And the last that we have, do I have black on here? Yes, I do. The last that we have is ATP that is produced. So from 1 pyruvic acid, we've gotten a lot of energy that's going to be able to be used in the electron transport chain. So let's go to that electron transport chain in the next screencast.